from partial linkage to genetic mapping. Once Morgan had understood how partial linkage could be explained by crossing over during meiosis, he was able to devise a way of mapping the relative positions of genes on a chromosome. In fact, the most important work was done not by Morgan himself, but by an undergraduate in laboratory, Arthur Sturtevant. Sturtevant assumed that crossing over was a random event, there being an equal chance of it occurring at any position along a pair of lined up chromatids. If this assumption is correct, then two genes that are close together will be separated by crossovers less frequently than two genes that are more distant from one another. Furthermore, the frequency with which the genes are unlinked by crossovers will be directly proportional to how far apart they are on their chromosome. The recombination frequency is therefore a measure of the distance between two genes. Comparisons between genetic maps and the actual position of genes on DNA molecules as revealed by physical mapping and DNA sequencing have shown that some regions of chromosomes called recombination hotspots are more likely to be involved in crossovers than others. This means that a genetic map distance does not necessarily indicate the physical distance between two markers. Also, a single chromatid can participate in more than one crossover at the same time, but that there are limitations on how close together these crossovers can be, leading to more inaccuracies in the mapping procedure. Linkage analysis usually makes correct deductions about gene order and distance. Estimates are sufficiently accurate to generate genetic maps that are of value as frameworks for genome sequencing projects.